Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Detection of antimicrobial compounds from culture. Supernatant using TLC bioautography. Table of contents. Introduction. Principle of TLC bioautography. Materials required with functions. Step-by-step procedure of TLC bioautography. Interpretation of TLC bioautography results. Advantages of TLC bioautography. Applications of TLC bioautography. Introduction. Microorganisms such as bacteria, fungi, and actinomycetes are well known for producing bioactive secondary metabolites. These metabolites include antibiotics, antifungals, antivirals, and other antimicrobial compounds that help the organism survive in competitive environments. During growth in liquid culture media, many of these compounds are released extracellularly into the culture broth, forming part of the culture supernatant after removal of cells. Detecting these antimicrobial substances is important for antibiotic discovery, natural product research, pharmaceutical screening, studying microbial interactions. One powerful technique used for this purpose is TLC bioautography, which combines chemical separation of compounds using thin layer chromatography, TLC, biological detection of antimicrobial activity using a test microorganism. This method not only shows whether antimicrobial activity is present, but also helps identify the number of active compounds, their relative mobility, RF values, their separation pattern. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Principle. Microorganisms synthesize secondary metabolites that may possess antimicrobial properties. These bioactive compounds are secreted into the culture medium during growth and accumulate in the culture supernatant. The supernatant is extracted using an organic solvent to concentrate these compounds. Thin layer chromatography, TLC, is used to separate the mixture of compounds based on polarity, solubility, affinity for stationary and mobile phases. Each compound moves a different distance on the TLC plate, forming separate spots. The developed TLC plate is then subjected to bioautography, where a test microorganism, indicator strain, is applied over the plate using an Agar overlay. During incubation, the antimicrobial compounds diffuse from the TLC spots into the agar. These compounds inhibit the growth of the indicator organism. After incubation, clear zones of inhibition appear at locations where active antimicrobial compounds are present. The position of inhibition zones corresponds to specific separated compounds. There are F values, helping in compound characterization. Download our microbiology study app made for researchers like you. Materials required with functions. 1. Culture and extraction materials. These are used to grow the producer microorganism and obtain antimicrobial metabolites from the culture supernatant. Microbial culture producing antimicrobial compound. The test organism, bacteria, fungi, or actinomycetes, suspected of producing bioactive secondary metabolites. Nutrient broth/suitable production medium provides essential nutrients for microbial growth. Specialized production media may enhance secondary metabolite, antibiotic synthesis. Centrifuge tubes, used to hold culture broth during separation of cells from liquid medium. Centrifuge, separates microbial cells, pellet, from the culture supernatant, which contains extracellular metabolites. Whatman filter paper or membrane filter removes remaining cell debris to obtain a clear, cell-free supernatant. Organic solvent, ethyl acetate/methanol/chloroform. Extracts antimicrobial compounds based on their polarity and solubility. Ethyl acetate is commonly used for moderately polar metabolites. Separatory funnel allows liquid-liquid extraction, separating aqueous supernatant from organic solvent layer. Rotary evaporator or water bath removes solvent to concentrate the extract, leaves behind crude antimicrobial residue. 2. TLC setup materials. These materials are required to separate compounds present in the crude extract. TLC plates, silica gel 60, F254. Silica gel acts as the stationary phase. F254 indicator allows visualization under UV light. Capillary tubes or micropipette, used to apply small, precise spots of extract onto the TLC plate. Developing chamber, a sealed container that holds the mobile phase solvent and ensures uniform chromatographic development. Mobile phase solvent system, mixture of solvents that moves up the plate by capillary action, separates compounds based on polarity differences. 3. Bioautography materials. These are used for biological detection of antimicrobial activity after chemical separation. Test pathogen, for example, E. coli, S. aureus. Indicator organism to test antimicrobial effect of separated compounds. Soft agar, 0.7% agar. Semi-solid medium that allows diffusion of antimicrobial compounds, growth of indicator bacteria. Nutrient agar plates support the growth of the test microorganism during incubation. Incubator maintains optimal temperature for growth of the indicator organism. Step-by-step procedure of TLC bioautography of culture supernatant. Step 1. Preparation of culture supernatant. Inoculate the producer microorganism into a suitable production broth. Incubate under optimal conditions, temperature, pH, aeration/shaking, if required. These conditions promote secondary metabolite production. After 24 to 72 hours of incubation, transfer culture into centrifuge tubes. Centrifuge at 8,000 to 10,000 revolutions per minute for 10 to 15 minutes. Purpose: centrifugation separates cell pellet, microbial biomass. Supernatant contains extracellular antimicrobial metabolites. Carefully collect the clear supernatant. Filter through Whatman filter paper or membrane filter. This produces a cell-free filtrate containing antimicrobial compounds. Step 2. Extraction of antimicrobial compounds. Transfer supernatant into a separatory funnel. Add an equal volume of organic solvent, for example, ethyl acetate. Why? Most antimicrobial metabolites are more soluble in organic solvents than water. Shake vigorously for 5 to 10 minutes. Allow the layers to separate. Collect the organic, upper layer. Repeat extraction 2 to 3 times for maximum recovery. Pool all organic extracts. Evaporate solvent using rotary evaporator, preferred. Water bath at low temperature. Purpose: removes solvent and concentrates bioactive compounds. Dissolve dried residue in 1 to 2 milliliters methanol. This is the crude antimicrobial extract. Step 3. TLC separation. Draw a pencil baseline. 1.5 centimeters from bottom of TLC plate. Apply small spots of crude extract using a capillary tube. Air dry the spots. Place plate in developing chamber with mobile phase. Allow solvent to travel 3/4 of plate length. Remove plate and air dry. Purpose: compounds separate based on polarity, interaction with silica, stationary phase. Step 4. Bioautography, Agar overlay method. Preparation of indicator organism. Grow test bacterium overnight. Prepare suspension approximately 10 to the power of 6 CFU per milliliter. Overlay process. Melt soft agar, 0.7%. Cool to approximately 45 degrees Celsius. Prevents killing bacteria. Add bacterial suspension to soft agar. Pour thin layer over developed TLC plate. Allow agar to solidify. Purpose: active compounds will diffuse from TLC spots into agar. Step 5. Incubation. Incubate at 37 degrees Celsius for 18 to 24 hours. During this time, bacteria grow to form a lawn. Antimicrobial compounds inhibit growth around active spots. Step 6. Observation. Observe clear zones against bacterial lawn. Each clear zone equals antimicrobial compound location. RF value calculation. RF equals to distance, moved by compound divided by distance moved by solvent front. Importance of RF: helps characterize compounds. Useful for comparing metabolites. Interpretation of TLC bioautography results. Clear inhibition zone on TLC plate. Confirms presence of an antimicrobial compound. Compound diffused into agar and inhibited test organism. No inhibition observed. No active antimicrobial metabolite present. Compound concentration too low. Compound not effective against selected test organism. Multiple clear zones. Indicates more than one antimicrobial compound. Compounds differ in polarity and mobility. Large inhibition zone. Suggests strong antimicrobial activity. May indicate higher concentration of compound. Small or faint inhibition zone. Indicates weak antimicrobial activity. Could be low concentration or partial inhibition. Zones appearing at different RF values. Shows presence of chemically different metabolites. Useful for compound comparison and profiling. No bacterial lawn growth on whole plate. Possible experimental error. Excess extract. Toxic solvent residue. Problem with bacterial culture. Interpretation of RF value. Each antimicrobial compound has a characteristic RF value. Same RF in repeated tests implies likely same compound. Different RF values leads to different metabolites present. Important concept. TLC bioautography confirms biological activity. It does not identify chemical structure. Further analysis required. HPLC, mass spectrometry, NMR spectroscopy. Advantages of TLC bioautography. Simple and cost-effective technique requiring basic laboratory equipment. Combines chemical separation and biological activity detection in one method. Detects biologically active compounds directly, not just chemical presence. Allows separation and identification of multiple metabolites in a mixture. Requires only small amounts of sample. Rapid screening method compared to advanced analytical techniques. Helps locate active compounds on the TLC plate using RF values. Useful in screening antibiotic-producing microorganisms. Supports natural product and drug discovery research. Suitable for preliminary analysis before HPLC or spectroscopic studies. Applications of TLC bioautography. Screening of antibiotic-producing microorganisms from environmental or clinical samples. Detection of bioactive secondary metabolites in bacteria, fungi, and actinomycetes. Natural product research for discovery of new antimicrobial agents. Analysis of actinomycete-derived metabolites, especially antibiotic compounds. Evaluation of herbal and plant extracts for antimicrobial activity. Comparative profiling of metabolites from different microbial strains. Preliminary assessment before advanced techniques like HPLC or LCMS. Studying microbial competition and ecological interactions. Quality control in research involving bioactive compound production. Academic and pharmaceutical research in early-stage drug discovery. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!